Normally the eye, the ear and the nose, so the sense organs and the cranial ganglia are very different from each other. Yet when we look early in development, it turns out that they all share a common history. Their identity is not fixed, so they don't know whether there are eyes, ears or noses. But if exposed to the right environment, they can give rise to all of these sense organs and cranial ganglia. The way we designed our study is we used different approaches to really try and understand what makes ear progenitors special, what makes them different from other cells. So we used a variety of methods, and all of these different approaches pointed to SOX8. We had no idea that SOX8 would be so important in ear development. SOX8 turned out to be one of the master regulators for ear formation. And what I mean by that is that it can drive cells that normally would never become the ear to become ear precursors. The ear is responsible for hearing and balance, and the inner ear has special cell types that are called hair cells. And these are essentially relay stations that transform the mechanical energy from the sound wave into an electrical signal. And this electrical signal is then transmitted to the brain. Now, when hearing goes wrong, that has many consequences. When babies are born deaf, that not only impacts on their ability to communicate, but also on their cognitive functions and social interactions. On the other end of the spectrum, during aging, most of us suffer from some form of hearing loss. And that is due to the death of hair cells. There are no drug treatments or stem cell approaches that will restore these hair cells. But in order to define these strategies for restoration and repair, we need to know how these cells normally develop, how they are normally set up. And we obviously are very excited to have found SOX8 as a potential master regulator, but we have no idea how it reprograms non-ear cells into ear cells. We do need to understand the molecular mechanism of how SOX8 works, and then we can use that to understand cell reprogramming in the context of the inner ear, but it might also give us some insight on how cell reprogramming works generally.